Our next speaker is Professor Sarah Jane Dawson, who is a clinician scientist and heads up the Molecular Biomarkers and Translational Genomics Lab at the Peter MacCallum Cancer Centre in Melbourne. So hi, Sarah Jane.
Hi, Anna.
Hi. Are you right to go?
Yes, I am. So thank you very much for giving me the opportunity to talk to you tonight. So I'm going to give a different angle on clinical trials and really just share with you what I believe is the link between discoveries in the lab and new treatments for patients. So as Anna mentioned, I'm a clinician scientist. So what does that really mean in practice? I'm a medical oncologist. So my clinical work, I'm looking after patients with breast cancer day to day. But the bulk of my time I actually spend as a researcher, as a scientist, and I lead a research lab that is endeavouring to make new discoveries that hopefully one day we'll implement into the clinic for the benefit of breast cancer patients. And I feel really passionate about what we call translational research. Traditionally, that's thought of as, bench to bedside research, where we make a discovery in the lab and then translate that into the clinic. But it works both ways. I guess, when you work in the clinic and you're looking after patients with breast cancer, you have an acute awareness of what we're doing well and where the gaps are, where our limitations are, and where we need to do better if we're going to improve survival outcomes from this disease. And I truly believe that clinical trials are the link. They're certainly the link between the clinic and the lab in being able to identify challenges and problems that we have in the clinic and take those problems to the lab to try and find discoveries, and vice versa. They provide an avenue for discoveries in the laboratory to really enter the clinical arena to benefit patients. And when we think about clinical trials, I guess the traditional model that we think of is clinical trials around developing new treatments. And by far and away, developing new therapies has been the backbone of clinical trials over many decades now and will continue to be so. But as Christabel alluded to, I guess we run clinical trials in lots of different areas to innovate in different ways. I've just alluded to what we call biomarker discovery here. Biomarkers are tests that we can use in patients with cancer to try and help obtain additional information. It may be additional information to help diagnose the cancer to begin with, to forecast how aggressive the cancer may be, and also potentially predict how well a patient will respond to treatment. But when we're developing new biomarkers or tests, we need to be able to validate those in clinical trials to show their value. In addition, clinical trials can also be used to improve tools for evaluation and monitoring of disease, such as MRI, as Christabel alluded to, and also monitoring responses to therapy. So I just really wanted to give you a feel for the fact that clinical trials can come in lots of different varieties, and breast cancer trials are certainly involved in running many different studies. I'm going to talk to you a little bit more about biomarker discovery today and clinical trials in this space, because that's certainly the focus of my research. And I guess when we're thinking about developing new biomarkers or new tests that we may be able to use in the clinic, it's not too dissimilar to, I guess, the road in developing new therapies for patients. Often that road is very long. It certainly starts in the laboratory where a discovery is made, whether that be for a new therapy or a new biomarker or test that we think might be helpful for using in patients with cancer. Then we need to develop that test more. We really need to understand how that assay or test is going to work and what samples we're going to be analysing in that process. Then we need to validate that test. So we need to make sure that the test is accurate and it's standardised and it can be done in a reproducible way. But then the most important step in seeing this biomarker reach the clinical arena is clinical trials. Clinical trials are really needed to prove the benefit of that new test, and without this, we will never see clinical adoption of either new tests or new therapies in this context. So the particular biomarker area that I work in is in the development of blood-based tests for patients with cancer. And I'm just going to spend a couple of moments talking to you about this area of research. So essentially it's looking at measuring something that we call circulating tumour DNA. And essentially for patients with cancer, particularly for patients with breast cancer, we're very reliant on obtaining tissue biopsies from patients to understand the features of an individual's breast cancer to plan their treatment effectively. And tissue biopsies are certainly important. They're never going to be replaced. But from a patient perspective, they can be very difficult to undergo. Certainly it involves usually a needle being put into the cancer, and that can be a painful procedure. Depending on the location of an individual's cancer or disease, particularly when it's spread to other areas of the body, biopsies are not always easy to obtain. And certainly they're difficult to repeat regularly during someone's treatment. And we know that the cancer is not static, it can change over time. So it can be very difficult to subject a patient to regular biopsies during the course of their treatment. The final limitation with biopsies is we know we're only getting information from the area of the cancer that we're actually subjecting to the biopsy. So that might not necessarily be representative of all of the changes in the cancer as a whole. So could we potentially do better with a blood test? Well, we know that in all of us, we all have small fragments of what we call circulating DNA in our bloodstream. And that DNA is being shed from healthy cells in our body. But in cancer patients, we know that they have much higher levels of this circulating DNA in their bloodstream, and that's because the breast cancer cells themselves can release these very tiny fragments of DNA into the bloodstream. We now have very sensitive and sophisticated methods for measuring this circulating tumour DNA in the blood. And it can all be obtained from a simple blood test. So most of our patients are having blood tests regularly during their treatment, and it's something easy that we can repeat over time to obtain additional information about the cancer. The other thing that's very exciting about these tests is that they can be really utilised in multiple different areas of breast cancer management. So we could think about potentially using these blood-based tests to help with screening or early diagnosis of breast cancer. Once someone's diagnosed with breast cancer, could these tests help us to understand risk and the prognosis of that individual from their breast cancer? When individuals are having treatment for breast cancer, can it help us monitor the response and understand how effective that therapy's been? At the completion of treatment, we want to know whether the disease is truly gone or whether there could be residual disease left behind, because that may place that individual at risk of relapse. So these tests may be very helpful in guiding that. And if we know that information, then hopefully we can offer additional treatment to try and counteract that residual disease. And finally, in patients who are unlucky enough to have developed advanced or what we call metastatic disease, these tests can be very helpful in obtaining additional molecular information about the cancer so we can hopefully plan treatment options more effectively. And the reality is, whilst these blood tests are under development in each of these different clinical areas, we actually need clinical trials in each of these different domains to prove the worth of these tests if they're going to be routinely adopted in the clinical arena. So this would take multiple clinical trials to see this be implemented at all of these different stages of disease. And I'm just going to highlight one clinical trial for you today, and it's looking at the far end of this spectrum where we might be able to utilise these tests for what we call molecular profiling or obtaining additional information about one's cancer to be able to plan therapy. And this clinical trial is called the CAPTURE clinical trial. It's led nationally by the Breast Cancer Trials Group. The particular patients that are eligible to enrol in this trial are patients who have advanced or metastatic breast cancer and a particular type of breast cancer, a breast cancer that's estrogen receptor positive and HER2 negative. These patients undergo a screening test with a simple blood test, and we're looking for the presence of circulating tumour DNA and a particular feature in terms of we want to see whether this particular breast cancer harbours a mutation in a gene. Traditionally, we would obtain this information from a tissue biopsy, but we're now able to characterise this from a simple blood test. Around 40% of patients with estrogen receptor positive breast cancer will harbour this particular mutation or change in their DNA. So we can pick up this change in around 40% of patients that have this test. If we find that information, then these patients are eligible to go on the trial, and the trial is really randomising patients and comparing treatment with a targeted therapy, a drug called alpelisib, versus standard chemotherapy. And essentially this trial is utilising a blood test to guide treatment with a new targeted therapy to see if we can really improve outcomes with this approach. So it's using a novel blood test to try and guide the use of a novel therapy and really target the use of this therapy to patients who are most likely to benefit. So just finally, I just wanted to summarise by just emphasising why we need clinical trials. I think they're really essential to prove the benefits of new discoveries, whether that be new therapeutics or new biomarkers, as I've alluded to. I think they're also very important for providing early access to new and improved tests and therapies for patients. And they're critical in order to change practice. Without a clinical trial, we will never see the adoption of these new tests or therapies into routine practice to be able to benefit patients nationally and globally. So they're really an essential part of driving discoveries into the clinical arena. Thank you.
Well, thank you, Sarah Jane. That was really interesting. When do you think you'll see results from CAPTCHA?
Yeah, so CAPTCHA started in the middle of last year, so I guess we're getting close to around 12 months. We're expecting the recruitment to the trial to happen over a 2-year time frame. And I think patients will stay on therapy for, ideally around 12 months. So I guess it'll be a couple of years before we see the readout of that therapy. But it's one of the only trials internationally to be using a blood test to be able to identify patients who may benefit from this new drug.
Yeah. All right. Well, we've got a couple of questions for you that have come in. So we've got one from Terry, and Terry has asked, what are the new technologies in breast cancer detection which will help improve outcomes?
So that is a really, really good question, and actually it marries very well with the blood-based tests that I've talked to you about tonight. So as I highlighted in one of my slides, one of the areas where these blood-based tests or circulating tumour DNA is being investigated is actually for screening to help with early diagnosis. So these tests are improving all the time. They need to be obviously very sensitive and specific to be able to assist and diagnose a cancer earlier. But that's certainly the direction these tests are heading. And, it's likely that we'll see these tests continue to improve for screening purposes and early detection in the next few years.
Yeah. All right, there's a question from Mary, and Mary has asked, how successful are immunotherapy trials for triple-negative breast cancer?
So that's also an excellent question, and I guess clinical trials have been instrumental in driving new treatments such as immunotherapy forward in the clinic. So essentially immunotherapies really work on the principle where we're trying to harness a patient's own immune system to fight against the cancer. And traditionally breast cancer hasn't been as responsive to immunotherapies as other types of cancers. But the one exception to that is really triple-negative breast cancer. There's a lot of research in this area currently undergoing, and I guess the major focus of that research is looking at how to combine immunotherapy with chemotherapy drugs to make the immunotherapy more effective. And also, are there biomarkers or tests that we can use to predict which patients may be most likely to benefit from immunotherapy? So which triple-negative breast cancer patients may be most likely to benefit from immunotherapy? So I think there's lots of progress in this area. We're starting to see these drugs being more effective when they're used in combination and trying to understand more about predicting which patients are likely to benefit.
You mentioned that immunotherapy has done quite well in other different types of diseases and other different types of cancers. Can that happen where you find out or have a breakthrough in a particular disease setting or a particular cancer, and then we can also test those treatments for breast cancer?
Absolutely, and I think we're translating knowledge from other cancers all of the time. So melanoma is a good example where immunotherapy has been very successful, and we've learnt a lot from those trials that can be, applied in the context of other cancers such as breast cancer. And the same applies outside of therapy. So with blood-based tests such as the ones that I've talked to you tonight, they have application across a whole range of cancers, not just breast cancer. So what we learn in the setting of one malignancy can definitely be translated into a breast cancer context.
Well, Tanya has asked a question about adjuvant therapies and what research is happening in this area. So maybe we need to start with what are adjuvant therapies and how does clinical trials research come into play here?
So adjuvant therapies are traditionally therapies that are given to women with early stage breast cancer after they've had surgery to remove their breast cancer. Adjuvant therapies such as chemotherapy and targeted therapy are then delivered to reduce the risk of the cancer returning, so to reduce the risk of relapse. Adjuvant therapies, particularly chemotherapy, have shown to definitely improve outcomes for women with high-risk early-stage breast cancer. And there's been a lot of change in this field over many years, I guess, through lots of clinical trial work that has been done in that space. And clinical trials here continue both to develop new adjuvant therapies that we think are going to be more successful, to plan the duration of those adjuvant therapies. We're also looking at better tests to understand how intensive that adjuvant therapy should be. So at the moment, many patients are delivered a very similar adjuvant therapy schedule, but it may be that we can utilise better tests or biomarkers to predict which patients need more intensive adjuvant therapy and which patients actually don't need additional adjuvant therapy. So there's innovation and clinical trials looking at the type of adjuvant therapy, duration, and how we can predict which patients really need it.
Yeah. It's interesting listening to you, Sarah Jane, because you're a medical oncologist and you see patients face-to-face in the clinic, but you also work in the lab. And I'd be curious to know what excites you about the future of breast cancer research and where it's heading.
I think that we've certainly come a long way over the last few decades, but we have a long way to go. But what I feel most excited about is that the pace of which research is happening now is ever increasing. I think with the plethora of new technologies we have available, we're actually able to make discoveries at a much faster pace, see clinical trials actually take off much more quickly, and ultimately that's going to lead to the integration of these tests and therapies much faster than what we've seen happen traditionally 10 or 20 years ago. So I think new technologies and being able to translate those discoveries more quickly is really what excites me going forward.
All right. Well, that's really great information. Thank you very much, Sarah Jane, and I hope it's not too cold in Melbourne.
It's not too bad today.